So the last part of this chapter is probably the most challenging, and we're really talking about what's happening with the different lab analysis techniques. And we're going to focus on describing some techniques. We're going to see these same techniques of chromatography and spectroscopy used in later chapters with other things besides drugs. And so we want to kind of go through, what do we mean by chromatography? What do we mean spectroscopy? And then we're going to talk about a few specific chromatographies and spectroscopies that are used in drugs. So chromatography is the idea of how to separate a mixture. So many times we don't have something that's pure. We have a mixture of different compounds, and I want to separate them so that I can analyze each compound separately, or at least determine what is in, how many components and what they are. So it's always going to be some way of separating the mixture. And we always generally have two different phases. One phase that has one property, and one phase that has a different property. And the idea is I'm separating it based off the preference for that particular property. I know that's very vague, but we talk about a stationary or fixed phase and then a moving phase. And the idea is, will the component move with the moving phase, or will it stay with the stationary phase? And depending on the multiple types of chromatography, these specific phases change. So the first type of chromatographies we have are thin layer column and high performance liquid chromatography. A thin layer really kind of helps me separate dyes, and it can be run off of a paper type of setup. And a column chromatography, I'm going to have a column of a specific substance that I pour my mixture through, and how it travels through the column will affect things. Very similarly, high performance liquid chromatography, also known as HPLC, will do a similar thing. And these are all based off of solubility in the mobile phase. I'm going to put some kind of mobile phase in there, and if it's soluble in it, it will travel with the mobile phase. If it's not very soluble in it, it will stay behind. And so the time at which it comes out will be based off of how soluble it is. And then I have gas chromatography, and that one is based off of boiling point. Things with low boiling points will separate out first, and then the high boiling points will come out much later, and they're going to be carried by the gas as the mobile phase. So when we look at this in general, let's get an example of, we have some different upper phase and lower phase. This is an example of a gas chromatography. So I have a liquid down here and a gas up above, and I have air moving along. And as I go through, notice we see that the blue ones seem to have a little bit more preference, probably a lower boiling point. And so I'm seeing them sort of preferentially either in the liquid or in the gas, moving towards the front. And by the time I have finished, I have a separation. The blue ones are up front and the green ones follow behind. And this is generally what we see. So, there's all sorts of factors and ways that we can improve the separation. That's really more for chemistry majors. But in gas chromatography, we talk about, and we're going to use, this one is very commonly used with drugs. So we're going to get into a little more detail with the gas chromatography. The carrier gas flowing through the column is the mobile phase, and then the stationary phase is a thin film of liquid that exists in the column. And lower boiling point components are going to move through very quickly. So they're going to come out first. We also talk about retention time. How long was it retained in the column? So we inject it in at one end of the column, and we're looking at how quickly it takes to exit the other end of the column. So the smaller the retention time, the lower the boiling point. Ones that are very high boiling point are going to stay in the column for a long time, and they'll have a longer retention time. So know that retention time, it would be measured in minutes or seconds, is going to be associated with short retention times or associated with low boiling points. Long retention times are associated with high boiling points. And we'll see that we end up with a spectrum. So here we have a carrier gas that's going to be flowing across the whole thing. Here's where we have an injector. The sample gets injected in, and here I have this column. So it's this column that's very hot, and it's got gas flowing through it. So as the low boiling points boil fast, and they're going to kind of come out of the column first, and the high boiling point ones will come out later. And then I have a detector that can kind of detect when chemicals are coming through, and a recorder, and I end up with this spectrum. So what I would see is this is kind of going along with time. So this component came out very early, has a low boiling point. This component came out much later, it has a high boiling point. And I can actually classify, if I run standard materials and say, based off of the retention time, I can tell which kind of chemical it is if I run specific conditions. So here would be an example of a mixture of barbiturates. So again, the bottom one, I do have to do some kind of a comparison. So the bottom one, I run a known mixture of barbiturates. So I know I've got this butabarbital, amabarbital, pentobarbital, and secobarbital, and phenobarbital. And I know from previous experience which peaks are which and how long the retention time is. And then I run the sample of my unknown, and I can look for similar retention times. So I see this peak right at a little bit, maybe 3.6 minutes corresponds to phenobarbital. And then this peak here corresponds to secobarbital. I don't see any peaks right around 3, so I don't have amobarbital in it. I probably have some butabarbital in it. It's kind of hard because this is an example of a peak that's going too high, so I really don't have a lot of resolution on it. But again, I can compare this and match up, and this is how we identify different drugs. And we can compare the different spectra. We call these a spectra, or spectrum would be the singular, and spectra is a plural. We also have thin layer chromatography, and this one uses a glass plate with a coated mixture on it. And what we see is that we put it into a liquid that something liquid that's kind of easy to evaporate and moves up by capillary action. It might be water, might be ethanol, and as the ones that are more soluble in the water or ethanol will travel up with it. Many times you'll see, sometimes we do color chromatography with kids with coffee filters, and you can separate the components of a marker, for instance. This would be an example of thin layer chromatography. So in professional thin layer chromatography, you're not dealing with dyes where they have a color to them. They're all colorless, but they tend to be active under ultraviolet light, and so sometimes they we'll use an ultraviolet light to be able to see the different spots on it. So the distance, how far it travels compared to the solvent, we measure it with a RF value for ratio of fronts. And this would be so rather than retention times in TLC, we'd be measuring RF values. So this is an example of what it would look like. You put a sample on here, you put it vertically in this column. At the bottom, I have the liquid, that's the mobile phase. And as it travels up, I'll get different spots on my plate. So those are chromatography or separating methods. Another type of general method is spectrophotometry. And in these, I'm going to see that different compounds absorb different characteristic wavelengths of light. And just as that substance can absorb the visible light to produce color, I also can see this with non-visible ones like ultraviolet or infrared. So it doesn't have to necessarily be visible, it's also invisible radiation wavelengths of light. And we can measure how much of that wavelength at different wavelengths that the compound absorbs. And that's very related to the type of compound it is. So the big ones we will see are infrared or IR and UV vis for ultraviolet visible. These are the ranges, and these correspond to different kinds of chemical behavior and very detailed identification. Generally, so you have to have something that's going to produce this light of different colors. You need to have something that's only going to allow one wavelength of light to go through at a time, the sample, and then something that can detect how much of it was absorbed or not absorbed, and then something that's going to actually produce a record of it. So this is kind of what an example might look like. Here's my radiation source. It may be a light bulb, it may be something else, but something that's going to produce a lot of different wavelengths of light. and then I have something that will split it up. So in this case, it's a prism that splits it into many individual wavelengths, and then I have a slit that says I'm only going to let one color through at a time. And we'll change that slit so that I can do this for many different, but I'm going to run it for one color, then for another color, then for another color, and then accumulate a whole spectrum. So it passes through the sample, and if the sample can absorb that wavelength, it will. If the sample doesn't have a chemical that absorbs that wavelength, the light just passes through, at which point this detector is going to measure it and say how much of this got absorbed or how much didn't get absorbed. And from that, I end up with a recorder. So for each wavelength, it measures the absorbance and puts it on a graph. I'm going to then change the wavelength by changing the slit, and do it again, and do it again, and do it again, till I end up having a spectrum, or a absorbance spectrum, which is like a graph with different wavelengths and saying how much does it absorb at different wavelengths. So let's look at what that would look like. So the UV and the IR spectrophotometry change in what kind of light am I putting in there. So infrared is associated, it's fairly low energetic. You know, we've seen it with TV remotes, has infrared, and we have infrared cameras, or that night vision type of cameras. And then we have ultraviolet, that's the kind of radiation we get a lot of out of the sun. So there are two different wavelength regions that can produce these, and so that's what changes. Now, of course, when you change that, you have to change how you're making that. Let's go back and look at the picture before. So the source would change, and the detector has to change. So they're different equipment, but the same general principle. And the UV spectrum is a very powerful tool that can pretty much identify what kind of compound it is. Not necessarily definitive. The IR spectrum is far more complex. It has a lot of peaks in it, and that can make it kind of difficult. So we call this a fingerprint method, because I get a very specific analysis off the IR spectrum, and I can tell one compound, slight compounds difference, I can differentiate with the IR spectrum, but it's also fairly complex to read. This would be an example of an ultraviolet spectrum. So notice on the bottom I have wavelengths. So these are different colors. So this might be one color to a different color. And I see that at different wavelengths, heroin can absorb right at around 250, 60, 70, around 280 wavelengths, nanometers of wavelength. Whereas amphetamine is a little bit closer to 250, and I actually see a couple of peaks here. So I can have a library or database that says I know which wavelengths amphetamine can absorb, and I know which ones heroin can absorb, and by looking at these I can identify which compound I have. But it's fairly simple and nice, but it's not as specific as infrared. Now, mass spectrometry is another very powerful and precise method, and in it we basically take the compound and energize it with some high energy electrons, and then fragment it into multiple different pieces. And from measuring the mass of those pieces, I can kind of do a jigsaw puzzle and determine what was the original compound. Different compounds are going to fragment in different patterns, and that pattern is very unique and fingerprint-like. So no two substances are going to have that same fragmentation pattern. So this is wonderful, but we see that this is an example of heroin on the top and cocaine on the bottom. And I get lots and lots and lots and lots of peaks that are characteristic. And these are assigned. We know what these different mass fragments are sort of related to in the heroin compound. But notice how complex these are. So this becomes a problem when we have a mixture, because if I have a mixture and I try to put this and this on top of each other, it would be a mess. But it is very precise, so it gives me a nice characteristic identification, but it's complex. So this is what it might look like. So it's a very similar type of thing. In some ways it's similar to the GC, only I no longer need to have a light source and a light detector. Instead, I inject it through, and this is preparing the sample and making the different, now I ionize it and I send it through this, basically this filter that can detect and make fragments out of it. And then I have a detector on the other end. And here I get my system of what we're looking for. So in this case, I've prefaced this with a GC. So this is a gas chromatographer, and then this part is the mass spectrometer. And we'll see that that's a very powerful combination. Again, what the problem with mass spec is, while it's a very precise identification, it's such a complex pattern that if I have a mixture, the spectrum becomes too complex. But if I can first separate the components and then do mass spectroscopy, that becomes a powerful combination. And so very often we'll refer to GCMS, or gas chromatography with spectrometry. So I'm going to take two machines and put them side by side. I will begin with a gas chromatography column, and that separates the components. So now only one at a time is going into the mass spectrometry, because remember, we have different times at which it comes out. And now at a given time, now I can do a mass spectroscopy of just that one component. And this gives me a very nice distinct pattern where I can really identify a whole compound, a whole mixture of compounds. So again, we kind of see what my spectra looks like. So I end up having many different spectra. So A might be an early spectra for the first component, B would be the second component that comes out, C. So I end up with a sort of three-dimensional. I have a time-based, A, B, C, D would be all at different times, and for each time I get a different mass spectrum pattern. And we can use different, what we call libraries, to help identify the different types of components that are in there. And first, so the gas chromatograph is separating them into the different components. So I have my A, B, C, D components coming out, and then they're each fed into the mass spectrometer, and I get multiple spectra for them. Remember, spectrum is one graph, spectra is two graph.